Everyone, class 12, subject biology, chapter principle and process of biotechnology. Biotechnology. Next, chapter 11 था. Next है component of a plasmid cloning vector, read in previous class. How an alternate selective marker and cloning vector for plants and animals, read in previous class. Let's begin. Start. Component host component host refer to the bacterial cell that have the ability to take up vector containing recombinant DNA. Method of introduce recombinant DNA. Component host cell are treated with divalent cations. These cells are incubated with recombinant DNA on ice, followed by heat soaked, then putting them back on ice by this bacteria able to uptake recombinant DNA. Microinjection recombinant DNA directly inject into the nucleus of animal cell. It is called microinjections. Biolistics या gene gun. Cells are bombarded with high velocity microparticles of gold and tungsten coated with DNA method. It is known as biolistics. Disarmed pathogen vector in case tumefacients and retrovirus. Next, process of recombinant DNA and technology. Isolations of genetic material DNA, cutting of DNA specific location, amplifications of DNA of interest using polymerase chains reactions, insertions of recombinant DNA into the host cell, obtaining the foreign gene product. First, isolation of genetic material. Isolations enzymes, cellulose, chitinase, lysozyme, ligase, etc. are used to extract DNA from the desired recombinant organism. For the process must be available in its pure form. First of all, cells are treated with a specific chemical to break open the cell to release cellular components such as DNA, RNA, protein, etc. Contaminants such as RNA and protein are digested with the help of additions of chilled ethanol precipitates out the purified DNA. Collections of fine thread in the suspension. Cutting of DNA specific location. In this method, a specific restrictions enzymes are used. These enzymes work as a knife and cut the DNA sequence at a specific site in such a way that fragments with desired gene are obtained. Isolation desired DNA fragment is carried out using agarose gel electrophoresis. Amplifications of gene interest is done using polymerase chain reaction. Obtain hybridized genes of interest with vector with help of enzyme DNA ligase. Polymerase chain recombinant DNA, shown this the figure, DNA. Several identical copies of can be synthesized in vitro. Two sets of primer chemical synthesized and oligonucleotide structures that are complementary to a region of DNA. The enzymes of polymerase primer using nucleotide provide in the reaction with genomic DNA as a template. The process of replication repeated many times segment can be amplified. 1 billion copies are made. Such as repeated amplification is achieved by use of the thermostable DNA polymerase bacterium tag Thermus aquaticus. PCR consists of three steps: denaturation, annealing, extension. Denaturations. Denaturations means double helical DNA denatured by providing high temperature. DNA polymerase does not get such high temperature since the DNA polymerase used in this reaction is thermostable from thermophilic bacteria Thermus aquaticus tag. Extension. Formation of complementary strand with the help of polymerase is called extension. Annealing process two sets of primer hybridized are hybridized to the separate single-stranded DNA polymerase. Insert the recombinant DNA into component cell. Insertions of recombinant DNA host done by several method transformations in case of bacteria, disarm vector biolistics and microinjection in case of plant and animals. The cell bearing recombinant DNA selected because the recombinant DNA exclusively have selectable marker present in them. Antibiotic resistance. Obtaining the foreign gene product. This is the stage for which recombinant was produced. Cell containing recombinant DNA will produce protein product. For large scale productions of the desirable product, antibiotic, vaccine, enzymes, etc., optimum conditions are to be provided. Continuous culture used to culture media drained from one side and fresh culture media is added from the other side. Cells are kept throughout in their exponential phase. Larger biomass produced by this method leading to higher yield. Bioreactor large vessels in which large volumes 100 to thousands liters of culture can be produced. Optimal growth conditions for microbes are present temperature, salt, vitamins, etc. Thank you class. This topic is completed.